So to kick off, hopefully, we have Victor. There's Victor. Victor Greiff, Associate Professor for Systems Immunology in the University of Oslo, Computational Antibody Profiling from Binding to Developability Design.
Hello, everyone. It's great to be here today. I'm going to start off the AI session. And just before, so in the morning, we heard already that AI might solve all of our problems. So maybe, yeah. So my talk will both revolve around antibody binding prediction and antibody developability prediction. The first part will be about binding prediction and the second part about developability. And just framing from a machine learning point of view, what do we mean by binding prediction? So if we have an antibody and an antigen, we would like to predict, ideally just based on the antibody sequence, what the antigen is, and ideally also the epitope. And one can also see this problem from the other side. Given an antigen sequence, we would like to predict all the antibodies that can bind this antigen. And there are challenges with that in terms of antigen binding prediction, epitope prediction, and antibody structure prediction. And from the developability side, the big problem of optimizing developability and predicting developability is that all of the biophysical properties, viscosity, solubility, aggregation, half-life depend on one another. And they're unfortunately, many of them are clustered also in the CDR3, which is also important for antigen binding. So once you optimize binding, you might disrupt developability and vice versa. And I think that's an ongoing problem in the field, how to multi-optimize an antibody. And in order to tackle this problem with statistical, mathematical, and computational approaches, there is a challenge of generating large-scale training data. And I'm going to go a bit into detail what I mean by that. And also, as I just said, the interdependence of these parameters really hinders machine learning approaches so far. So when we, of course, one immediate goal is binding prediction. So again, given an antibody, I want to predict the antigen or the antigens, because antibodies can react with multiple antigens, and the other way around. But beyond binding prediction, what we actually want, because many of us are biologists, not just AI people, we want to understand the underlying rules of binding. So what actually makes this antibody bind this antigen and vice versa? And by rules, there are many ways to define this, but one way to defining this is we can define this in terms of the sequence-based motifs and the non-continuous structure-based motifs that actually mediate the binding landscape between antibodies and antigens. And this is actually what we want to learn from training data. So what we would like to have in the not hopefully too distant future is just like ChatGPT can give us now text, we would like to have a Rosetta Stone where you just input a bunch of sequences, antibody sequence, and it gives you all the antigens that these antibodies can bind and vice versa. And we are still a long way ahead to having this in hand. But there are novel experimental techniques that have been published in the recent past over the 4, 5, 6 years that can resolve the antigen binding landscape at fairly high resolution. So we have, of course, LibraSeq. We have then the supercharged LibreSeq, which is the Beam app platform from 10x, and we have also deep mutational scanning platforms that can really profile at large scale the mutational landscape and how one or a few mutations can change binding at very high resolution. And so one problem that we saw when we worked, when I started my lab in 2018, we had this idea of, can we learn some kind of motifs that are universal to antibody-antigen binding? So we looked at, back then, and it was before COVID, so we had about 800 non-redundant antibody-antigen structures. And we looked at, is there a common vocabulary across these very different antibody-antigen structures that link them via a common vocabulary? And we indeed found some motifs that are repeatedly found across very different antibody-antigen structures. And from that, we could kind of infer that there must be some underlying predictability in antibody-antigen binding. But of course, one cannot solve antibody-antigen binding with 800 crystal structures. And one can now either go two ways. One can wait 20 years until we have, hundreds of thousands of 3D structures, or you can approach this in a slightly different way, that you can develop synthetic or simulated data to mimic the structure space in order to already develop machine learning approaches now that you can then apply to experimental data once we have sufficient experimental data for learning. And this is what and this is the route that we took. And why is this route so important also? So there are many different machine learning approaches that one can take to antibody-antigen binding. And what you find in many papers is that if they have used machine learning approaches, these machine learning approaches are then tested in the experimental world with maybe less than 100 expressed antibodies. And of course, machine learning can generate hundreds of thousands of different variants that could be binders. So just from a statistical point of view, it is carefully phrased, not good practice to just look at a few expressed antibodies to rule out which machine learning approach is best for a given binding task. So what one needs is actually synthetic data at super large scale to kind of rank different machine learning approaches in terms of their usefulness for binding prediction. And this was also recently very nicely explained by this preprint from this group at Carnegie Mellon that explained in a very comprehensive way that if you have a computational approach that does a certain thing, your data needs to be ground truth in order for you to test whether your approach does actually the thing that you want it to do. And most of the experimental data that we have in hand is actually not ground truth because we do not know the generative rules of this experimental data. That means to really develop trustworthy computational approaches for biology and especially antibody design, we need both experimental and large-scale synthetic data for benchmarking. And this we outlined in this review that we published last year, where we kind of outlined that both the interplay of good real-world relevant synthetic data as well as experimental data can lead to actually develop good models that do not only predict well, but that are also interpretable and that can then help us to understand the underlying rules of binding or developability or any other rules. So last year then we published a simulation framework for antibody-antigen binding, which is called Absolute. I will explain it on the next slide. But what the simulation framework simply does, and it's the lowest hanging fruit that you can think of, and it's still a very high bar, is that with the simulation framework in the simulated world, you can rank machine learning approaches in terms of their usefulness for binding prediction. And then all we claim in this work is that this ranking will then transport to the real world. So once you have found that a given machine learning approach 1, for example, works best for binding prediction, this will also be number 1 on experimental data. And that's, of course, very useful because you don't have to test thousands of different machine learning approaches on your experimental data. So this slide is very complicated. It's just to show you that this simulation framework for antibody-antigen binding generates 3D binding paratope-epitope interfaces at the scale of billions. So just to put this into perspective, crystal structures we have in the magnitude of thousands, maybe 10,000s, and here we can generate billions of synthetic antibody-antigen structures with paratope, epitope, and affinity information. And this synthetic data is of course not native-like data. If we could generate native-like data, we wouldn't need machine learning. But it replicates many of the properties that antibody-antigen binding has. And one of the main properties that makes antibody binding prediction so difficult for machine learning that we replicate in the simulation framework is that what is shown here in a simplistic way is that, so here a point is an antibody and the color is what antigen it binds, is that points that are very close in sequence, here very, very close in this plot, can bind very different antibodies. And sequences that are very different from one another can bind the same epitope or antigen. And this is something that is very confusing for machine learning approaches to actually learn and to also generalize. Another great thing with synthetic data is that you can actually test whether your machine learning approach can understand the underlying rules of paratope-epitope binding. And here we define the rule or rules as paratope structure. And what I can see on this plot, and you would see if I explained this to you for an hour, is that you see this clustering here by the different colors. And that means that without having given to the machine learning approach any affinity information or structure information, very simple machine learning approaches with a binder-non-binder problem can already learn something about the underlying paratope structure, so which amino acids in a given CDR3 sequence are important for binding. And that's quite nice. What we had also seen, what is a bit not disturbing, but should lead us to think about how to set up machine learning problems. So many machine learning problems for binding prediction are set up in a way that you separate your data in binders and non-binders. And what we have seen in simulated data and what people have also seen in the TCR epitope prediction field is that how you define the non-binders can not only change prediction accuracy of your model, but also can change what you learn about the underlying biology of TCR or antibody binding. And we wanted to go a bit deeper into this problem, so we used again our synthetic data, where we can, and this you could never do with experimental data, where you can cut your data, and these are hundreds or millions of data points, where you can cut your data into binders, weak binders, and non-binders, and where we can change the definition as a non-binder as being a very bad binder, so a non-binder or a weak binder, so almost a binder but not a binder. And this is in terms of binding energy. And what you see here is how we define the negative training data, how prediction accuracy really depends on how we define the negative data. And that looks quite bad for this task where the non-binders are defined as weak binders. But what we can show, what I'm not showing here, is that when you want to generalize to a very different test data set, then actually this model that looks worse than the others actually generalizes much better than the models that looked much better from just looking on in distribution testing. And yeah, I think this is what I said. I think I can move to this slide. And another big problem is then how does the definition of the negative data set impact learning of the rules? So what we see is that, for example, if we define the non-binders as the weak binders, so the almost binders, then the model can learn quite well without us having given any affinity information, the affinity of the antibody sequences in a very good correlation. And this is the certainness of the machine learning model, and this is the energy, and there's a very nice correlation. And if you then quantify this correlation across the different antigens that we tested and the different negative binding definitions, then you can see that the correlation between how certain the model is of which amino acids are important for binding in terms of contributing to the binding energy and the definition of the training data, it really depends on how you define the negative training data. So for some definitions where we saw previously that the prediction accuracy is good, these models actually do not learn anything about the underlying biology of binding, here defined as which amino acids are important for binding, whereas some model setups learn this much better, here quantified by the correlation. Yeah, so it really, and this you can only see with synthetic data, and I think this really shows that we can use machine learning for not only prediction, but also biological rule discovery, but we need to set up those machine learning problems in the right way. And learning how to set up those problems, there really synthetic data can help. And then once we are sure of a given setup, then we can transport to the experimental data. So now I'm just going to show some work that we are also doing in our labs in our lab in terms of binding prediction. And here we are tackling a big problem of the antibody discovery field, where there are a lot of data sets where we only have the label on the repertoire level. So for example, there is a lot of public data out there, also in companies, where you only know that this antibody repertoire comes from a healthy control person or from an HIV-infected person, but you do not have the labels for the individual antibodies in this repertoire. You could also think this from the way of phage display. You have a control library and then selected libraries, but you do not know what the binding of the underlying clones is. So what we would like to do here is to only input into the machine learning approach, which is based on topic modeling, repertoire labeled data. So so we have only label for this bunch of sequences, but then we want to infer those sequences that are actually binding to the antigen, which are marked here in red. And I'm not showing you any data, but using both synthetic and experimental data, we can show that it works, not surprisingly well, that it works reasonably well from a given threshold onward. So you need a certain concentration of binders in your data set, and from that onward, it works reasonably well. So I think this kind of machine learning really tackles one of the big problems in the field, because we have a lot of data that is being unused for antibody discovery, and we need machine learning approaches that can use these repertoire labeled data to isolate actually the binding sequences in these data. And this approach can not only identify the sequences, but also generate new binding sequences based only on repertoire labeled data. I think we're going to preprint this in a few weeks. So building on what I showed you before is that how you define the negative training data set really can bias your learning. So ideally, you would like to learn the binding rules only from a binder data set, because then you don't have the bias of the negative data set. So this is what we try to do here, only learning paratope motifs from one labeled data set, so only from the binders, and using, and there we are a bit further away from publishing or preprinting, but again, we're doing this on synthetic data and also experimental data. And what we see, at least for now, on the synthetic data, and hopefully also on the experimental data, that we do not even need the negative training data. We can also, it is possible to just infer binding motifs from one labeled data set. I think that this also would free up a lot of pressure in the field of how you define non-binders. Yeah, so we're very excited about this project. And yeah, so this is based on, for the machine learning people, we are using a generative model here. So we can also generate new sequences, and for the interpretability part, we're using integrated gradients. And then, of course, you might think, okay, these are crazy people. They're only working with simulated data, and they do not know anything about the real world. But we're not that crazy, so we're also generating our own experimental data sets. And what we are very interested in is, because a lot of people either look at B-cell receptor sequencing or serum antibodies using mass spec for investigating the humoral immune response. But there is not so much work on actually combining bulk B-cell receptor sequencing for coverage of the diversity of the repertoire, single-cell sequencing for coverage of the paired chains, as well as mass spec to cover the serum antibody diversity. And in this project, which hopefully will also get preprinted quite soon, we are trying to, how can we combine all of these approaches together to really get a comprehensive understanding of the humoral immune response. And lastly, since we are very interested in mass spec, so obviously antibody mass spec has been started with 2 Nature Biotech papers in 2010, and there has been a lot of work on this, especially from the George Georgiou lab, which has analyzed antigen-specific responses using mass spec. But what remains a bit unclear in this field is what is actually the sensitivity of antibody mass spec in terms of what can we actually detect. So how low can an antibody be in a given serum sample that we can still detect it, and how does this detection depend on the proteases one uses, as well as the bioinformatic processing that one uses. So we generated, I think, one of the largest mass spec data sets, so more than 1,000 samples with ground truth monoclonal antibodies where we know the sequence. And then we spiked them in different concentrations, different mixtures together, and then applied different enzymes to them, and then wanted to understand how low in terms of concentration can one go, what is the impact of mixing different antibodies on the signal, and can we also use AI-based de novo prediction without a reference database of predicting back those antibody sequences. And to our surprise, de novo prediction works surprisingly well, but you need a higher concentration than you would need for a reference, for reference-based searches such as using MaxQuant or MS-Fragger. So I'm not showing you much data on that, but if you're interested in that, I would like to chat further with you. Something that, in terms of rule discovery, that we are really adamant about is, so how do you define rules? So here we teamed up with the linguistics department at our university, because because a lot of people talk about we want to understand the antibody language, right? Or we want to understand the protein language. But what does it actually mean? How do you define protein language, antibody language? Is it actually the same language as a linguist would define human language, German, English, etc.? So we teamed up with them, and they didn't know anything about antibodies before, but we had a wonderful postdoc, Mai-Ha Wu, who kind of dove into the antibody world and then tried to juxtapose natural language and antibody language, finding the commonalities and also differences between those two concepts, and coming up with ways that we now have a roadmap that we can use for encoding better the data that we input into machine learning approaches to make it more interpretable and maybe also increase prediction accuracy. So that would be the next steps in the future for this work. So just to summarize the first part, so our lab, but also of course many other labs, have shown that antibody binding prediction is possible, but the one caveat is we need large-scale data sets for that, and really domain-adapted approaches that are geared towards antibody binding prediction. And just to summarize the state of the art, I think a lot of people, at least in the structure space, work on paratope-epitope prediction. What we would like in the future is end-to-end antibody-antigen prediction without defining a priori what the paratope and the epitopes are. We would like to, especially from a pharma point of view, generate antibodies towards multiple targets or target specifications, and we would really like to understand from a fundamental point of view the underlying rules of binding, and especially for that we need benchmarking as well as synthetic data. I think I can skip that time-wise. So quickly, just focusing on developability. The field is also growing, so the developability field is growing in terms of predicting developability from the antibody sequence or structure. And there has been a lot of work by many different labs, also many great reviews. But one problem in this field is that people think that, or there seems to be a certain stream of papers that mostly focus on learning what is a good antibody based on the antibodies that have made it to the clinic so far. And that, of course, has the statistical bias of survivor bias, and you do not really know why these antibody made it that far, right? So you learn on a very small distribution and not the underlying distribution of all potential antibodies. And furthermore, it has been shown that many antibodies, so the more natural an antibody is, the better its developability is. So, and that has been also shown in multiple papers, and some companies are even based on that concept. So what we try to do is look at very large-scale data sets, compute many developability parameters on these data sets, and then try to see how redundant those are, how predictable they are, and how sensitive to mutation they are. Again, this is quite a large project, so I'm going to focus on some key points, but briefly we looked at experimental data, so more than 2 million sequences, but not only human data, also humanized mouse data, patent-submitted antibodies that was kindly provided to us by Natural Antibody, the company Natural Antibody, and then also therapeutic antibodies, and then we computed for all of these sequences structures, and then on top of this, the 80 developability parameters that can be categorized by these labels. And now, just one point that I think is very interesting. So when you take these 80 developability parameters, you can just do a PCA, and that spans the developability space. So in blue is this human VH space, and then we just projected on the space the humanized mouse, the patent-submitted antibodies, as well as the therapeutic antibodies. And you can see that most of those antibodies lie in the natural space, right? So, which is consistent with the hypothesis that we had, that most antibodies that are being developed lie actually or are close to natural antibodies. And here, when I was last year at Fair Journey, there was one comment in the audience that said, okay, but that could be biased by the physical-chemical parameters that you looked at. But when you use a protein language model, so here now this is the space of a protein language model, same projection, and then again you project on the blue space, so the natural space, the humanized mouse and the patent-submitted antibodies and the therapeutic antibodies, you can see interesting patterns that the humanized mouse does not fully cover the native space. So something in the VDJ recombination of this mouse does not seem to fully recover what the native space can give us in terms of antibodies. Whereas, surprisingly, the patent-submitted antibodies seem to be all over the place, which could just mean that people just submit whatever to a patent, right? So, and then, of course, we are undersampling on the therapeutic side. These are only 300 data points, and you can see that they are focusing on different areas and are, but there it's a sampling issue, so there I think you cannot say. But what one can say is that they're also inside the natural repertoire and not outside. So to summarize, before I go to 3 slides to a different topic, is I think as a roadmap for de novo antibody design, we need large-scale curated data of sequence structure and developability, domain adapted and task adapted machine learning approaches, large-scale experimental and computational benchmarking of AI approaches, academic and industry collaboration, and I think AlphaFold has shown us that we need open competitions to select the best machine learning approaches in an unbiased manner. Just need 3 more minutes. 2. Since I'm the chair of the AIR, so who knows what is the AIR community in this? Okay, that's 20% for the audience that's not here. So we are a society, and we are focused on developing standards for generating, curating, and sharing adaptive immune receptor sequencing data. And we have many working groups that work on different aspects of the standardization. And the interesting thing is that all of these data are curated in the same way. So we have standards, and these standards are called AIR compliant, both on the experimental side and the computational side. And the standardization really gives one the power to just read these into different computational tools, such as machine learning. And so I think as all of us as a community, we should move towards standardizing data input, data output, and how we curate our data sets. Yeah, and we have published a lot as a community on this topic. So, so we have a lot of standards for going from germline to computational approaches, and I think a lot of, so here I take the example of Fair Journey, but there are a lot of other companies that are making large-scale data sets, right? And many companies and also academic labs have the problem of curating their data in a way that it can then be merged into machine learning approaches. And I think if we all would invest in curating, standardizing our data sets using, for example, the AIR standard, we would I think, move closer and much quicker to the to solving the common problem of, for example, binding prediction or developability prediction. And we have seen the power of curation in the field of biology, how powerful this can be, because AlphaFold would not have been possible without curating data sets over decades. We have seen this power also in other fields such as ChatGPT, but also cancer prediction from images or chemistry prediction. And many of you have probably also seen, if you're on Twitter, that what can happen if data is not pre-processed in the way that it should be, because then machine learning approaches just pick up on artifacts that are just not biology, and then it creates to findings that are just that are not correct. Yeah, so I think curating in a consistent way is super important, especially as the data gets more high-dimensional. And the good thing, but also the bad thing of machine learning is that it can pick up on very small things. So if your data processing is not correct, it can pick up on these problems in your data processing step, and it can lead you completely astray. And with that, I would like to, last slide, just advertise the next Air Community meeting, which will be also in Porto here next summer. This was a picture from the last meeting where Dennis Burton was also the keynote speaker. So if you like antibodies and or TCRs, then you need to join this conference. And with that, I would like to thank my group and as well as you for your attention.